Hello, everyone. My name is Ekhtiari, and I'm gonna kind of coordinate the meeting with my co-chair, Colleen Hanlon. So we already have a lineup of something like 12 speakers right now. It's gonna be a delightful meeting discussing about optimization of brain stimulation interventions for people who are suffering from substance use disorder. And the basic idea is, as you can imagine, there are neural circuits involved with kind of addiction-related behaviors. And the question is there any chance that we can target these networks, these neural circuits with different non-invasive brain stimulation technologies? And we already have a list of different technologies that could be used as non-invasive brain stimulation technologies in addiction. But the question is how we can optimize those non-invasive brain stimulation interventions for people who are suffering from substance use disorder. And that is the main target of the webinar today. So we will try to basically discuss about this specific issue. And when we talk about optimization, we have optimization at least in 4 major dimensions. We have it in context, we have it as target optimization, dose optimization, and timing optimization. So we'll have it in at least four different topics. So
you want to put it on the holder.
you already have those.
I don't want you to get it caught on the cord. Thank you.
Yeah, it would be nice if people just kind of turn off their mics. Okay, good. So talking about these dimensions, we have kind of the contextual dimensions of optimization, and one of the contextual issues is about how we can provide people with a context that is related to their substance of use, and we know that probably drug use could be one of the potential sources for optimization in the contextual level for people. And we developed kind of a series of Q databases that people can use, and there are already some other databases available. And we recently have been able to put together a series of studies trying to see how we can basically use drug Q reactivity as a paradigm to provide a good contextual kind of background for people who are gonna be using brain stimulation interventions. And the good thing is that we have always been we already have been able to kind of put together a consortium of labs that are doing fMRI drug Q reactivity. We have already collected over 3,000 patients with fMRI drug Q reactivity data. So that is amazing that we are extending that basically Q reactivity initiative. And those who are interested, you can have access to that consortium. And we are also inviting others who already have fMRI drug Q reactivity data to join that consortium. I will discuss how this kind of fMRI drug Q reactivity databases could be used for developing new targets for non-invasive brain stimulation addiction. The next level that we will discuss today in the webinar would be target. So we will discuss a lot about target and how we can find the right target and how we can optimize the target in the group level and how we might be able to optimize target in the individual level. And there are 2 seminal works that are going to inform what we are doing in the field of addiction. One of them is, the Nolan Williams work that has been recently published. And people have heard about that. And the other is the seminal work that Michael Fox published recently. And these 2 works, I think they are going to definitely inform what we are going to do for target engagement and finding the optimum targets. And when we talk about targets, there are different targets that are potentially important for what we are going to do. As you can imagine, there are many areas or networks that people are interested to target, not only in addiction, but in other areas of psychiatry. And one of those targets, for example, is the frontoparietal network that we are interested to, and we have been kind of trying to see how we can engage frontoparietal network. But we realize that, when we go for classic F4, P4 montages, we do not have connection that we have been expecting for. And we need to use fMRI data to find the right and optimum montage or target to be able to find the right network to engage. And we realized that we basically need to go for F3 to CP5 and F4 to CP6 to target the actual frontoparietal network that is involved in drug cure activity. And with that optimized montage, we showed that we can basically engage the network that is interesting for us. Meanwhile, we showed in some of the new work that we are doing that there is a significant level of inter-individual variations in terms of the actual target that we can use. And there are always a discussion in terms of how we can take into account these inter-individual variations in the target that we are interested to. That's another discussion that we can talk about during the webinar. Meanwhile, we already have a discussion regarding the dose and how we can optimize the dose for stimulation. For example, for the same frontoparietal stimulation, you can see that there is a significant level of variation among different participants. So this is the histogram of participants that we had in a database of 60 patients with methamphetamine use disorder. And you can see that people would receive a lower level of current to higher level of current with the same montage, which is, I mean, interesting, but meanwhile frustrating because people would have different levels of electricity in their brain when we do the stimulation. So we need to think about how to optimize that in the group level and also in the individual level. And even when we change the montage, the distribution could be quite different when we use the same level of current in both sides. So we might even need to optimize the amount of current that we put in each side if you want to have a sort of a balanced stimulation in both sides. So there are lots of discussion in terms of how to optimize the dose in the individual level and also in the group level. And we recently published a paper discussing about the challenges of these optimizations. And the last dimension of optimization is about the timing. And there are many aspects of timing that should be discussed for individualization of non-invasive brain stimulation. One of the things that we do with frontoparietal synchronization, we always have an issue in terms of what is the right frequency of electrical stimulation and what is the right phase difference between the nodes that we are stimulating. And finding the right target within this large space of frequency and phase is something really complex to do. So we need basically to do one trial for each of these dots, which is really complex to do. So we have been able to put together another consortium of people who are doing electrical stimulation inside the scanner. And that is helpful to be able to basically have what we call closed loop TES fMRI. So we can put people inside the scanner, we can define a target, and we can try to optimize the stimulation parameters to be able to target at a specific network that we are interested to. We have recently been able to put together a full setting for closed loop TES fMRI, and we put together a recent protocol paper about the feasibility of this kind of new setting that could optimize the timing of the stimulation in an individual level. And this has been recently kind of accepted in Brain and Behavior, so it will be out soon. So these are the questions that we have and how we might be able to address optimization issues, specifically in the field of addiction neuromodulation. And our next speaker, Dr. Colleen Hanlon, who is one of the shining stars in the field, and my co-chair here, is going to give you an idea about how we can do optimization using provocation-based targeting. Colleen, please.
Great. Well, thanks so much. And again, thank you all for being here. And Hamed, your leadership skills are incredible, as always. So I just have 5 minutes. And what I wanted to kind of like close out here, because you all are, this is really a great forum just for kind of discussion and thought-provoking things, is what's the role of provocation, particularly for substance use? Weird, sorry. What TMS is most used for right now is depression, right? And depression, generally the assumption is that depression is a fairly stable behavioral and brain state. Whether you do the MADRS or the Hamilton or the PHQ-9 at 8 in the morning or 5 in the afternoon, the score is assumed to be fairly stable. Next. Thank you. And so what we see in sort of the TMS world is kind of the current trend is to do targeting using resting state functional connectivity. And the assumption there is that this is a picture of the brain in a fairly stable state. We can have a separate discussion on whether or not resting state is a stable thing, but that's kind of the idea, that somebody comes in, you take a picture of their brain in resting state, and then you use that as the way to figure out where you want to position your TMS coil. And you do it one time, and then you apply it for all sessions. Next. And so the presumed mechanism, therefore, is we've now identified this functionally guided target that is there, and it's present for these people for their, and it's just kind of a symbol or a biotype of their disease subtype, and we're going to manipulate it every day with TMS. Next. Addiction, however, is different, right? All of you guys probably interact with people with addictions in one way or another, whether it's clinically or in your research lab or in your family or community. In our studies, for example, with alcohol use disorder, most people that come in, they meet criteria for alcohol use disorder, but if you ask them, do they think about drinking at 8 in the morning, they say, oh, no, never. I can work, I go to work, it's all fine, but it's when I get home and I'm in a certain environment or I have a stressor, that then it's like this state kicks on and I begin drinking and I can't stop drinking. And so that's dynamic, right? If we did a craving-based image at like 9 o'clock in the morning, that's going to be a really different brain state than when ideally we could have an MRI scanner in their house, right? And we could take a picture of their brain there. Certainly EEG would be a really nice way to look at that. But because the nature of addiction is a very dynamic disease that changes out the kind of behavioral problems, like the actual drinking, for example, isn't continuous, whereas depressed mood tends to be a continuous thing. I think we need to think a little bit about these dynamics when we think about TMS treatment. And so the current trend in TMS treatment for substance use disorders, but actually this is also true for OCD, which has similar kind of fluctuations. People don't tend to engage in compulsive behaviors 24/7. It tends to be associated with an environment or a cue, is to provide a provocation right before the TMS delivery. Next. And the presumed idea there in these studies that are using a provocation, either a script or cues, is that we're priming the brain state. We're kind of calling it into working memory, perhaps. And then we're applying TMS in order to manipulate this state. Rita Goldstein and I have had some interesting conversations about whether this is kind of a memory reconsolidation mechanism or what's going on. I think there's a lot of work there. Next. And so just kind of very quickly, what we see here is that provocation is now part of the FDA-approved protocol for OCD. So there's always priming initially before the TMS is given. It's also true in the FDA protocol for smoking. There's always a smoking prime before the TMS is given. And both of these FDA pivotal studies were preceded by studies that evaluated whether or not the cue was relevant. And they both found that the effect size was greater when individuals were primed with the cue. So that's pretty cool, as Dr. Fox is going to tell you about probably in a bit. When we look at these coils, in both of these FDA pivotal trials, they use wide field H coils. And I think this might be an interesting way to think about it. So with these wide field H coils, we're not targeting in a very specific brain way. But when we have cast a wide field and we pull the brain into a specific state, then the electric field is more likely to overlap with the individual's kind of fingerprint of the brain state associated with smoking or the obsessive behavior. And there's 2 studies that have been done recently in alcohol use disorders as well, which use provocation. One was by our group, one was by Bumi Zanigan's group. And the studies in many ways were similar. They used different coils. They had all kind of targeting the medial prefrontal cortex, but slightly different areas of the medial prefrontal cortex. And kind of the summary here in just a brief amount of time is that in both cases, at the 3-month follow-up, individuals with alcohol use drinking or alcohol use disorder were drinking less, and they had less brain reactivity to cues. And in both cases, they used provocation. Next. Thanks. In one case, they used 3 minutes of holding and smelling the beverage, but not actually consuming it. So the person has the beer or the glass of wine right there in their hand while they're receiving the TMS. Next. And in the other case, in our case, we had people actually think about when was the last time you were drinking. And our prime was keep thinking about alcohol and negative aspects of how it makes you feel. So that brings up a lot of questions on what's the right prime. Do you have them think about the rewarding aspects of the drug? Do you have them think about the reasons for quitting, which is more of an executive control kind of strategy? I think we can probably learn a lot from our kind of clinical psychology friends in this regard as to what the optimal instruction might be. And then one last thing, Dan McCallie, who was a student in my lab, he's now a postdoc at Stanford, looked at the data from our biological psychiatry study, and what he saw, he divided people into groups that had either an overlap between the electric field, which is kind of in blue here, down in SIBNIBs, and the fMRI brain reactivity, in this case, alcohol cues. So people that had overlap, people that did not have an overlap. In this case, they generally, when they react, their brain turned on to cues. It wasn't in the same place that we were stimulating with TMS and sham. Next. And if you follow the purple line here, next. Next, sorry, next. So he divided it into groups. We had 50 people in the study, and next. 13 of them have overlapping fields, and if you follow the purple line here, this is the percentage of the sample with at least one heavy drinking day in the last 30 days. Next. And you'll see the purple line here on the bottom. So the people that had an overlapping electric field with their brain reactivity to cues, they had the best outcomes. So they were less likely to drink at 2 and 3 months than the sham group or the people that didn't have an overlap. And so that's kind of the thing I want to leave everyone with is that it is probably important, which kind of makes sense, that the electric field intersect the brain network that we're trying to alter. And in addiction in particular, but probably also OCD, maybe PTSD, these are sort of state-specific brain networks that aren't engaged all the time necessarily. They tend to be activated in certain environments or to certain cues. And so that's probably important for us to incorporate into our treatment. That's all.
Thank you very much, Colleen. Thank you very much. It's really an exciting talk and really kind of thought-provoking how to optimize stimulation based on the cue presentations and cue provocation. There are lots of opportunities on that line. Thank you very much. Now we are going to go for the next speaker, who is Dr. Michael Fox from Harvard. And Dr. Michael Fox, one of the pioneers of doing network-based or circuit-based neuromodulation.